Hi, my name is Swatantra and I am from the Life Sciences Research Group at TCS. Today, I am going to be sharing our work titled An Augmented Transformer Model Trained on Family Specific Variant Data Leads to Improved Prediction of Variants of Uncertain Significance. Let's look at what are these variants of uncertain significance, or in short, VUS. Any variations in a genetic sequence are classified as variants of uncertain significance when the association with the disease risk is unclear and the effect of these variations on protein functions is unknown. Now let's focus on several aspects regarding these variants, such as what's the importance of these variants, why in general these variants are difficult to predict, and why we need to develop reliable methods to predict the significance of these variants. Let's focus on why these variants are important. From the context of personalized medicine, it is a necessity to have a reliable method to classify these variants for clinical diagnosis, genetic studies, and protein engineering. Computational predictions for classifying these variants results in efficient and scalable preliminary assessments on how these variants affects human health. In general, these variants are difficult to predict because of several factors such as lack of functional data. The variants are found in less characterized domains, and impact of these variants in protein function is difficult to uncover. So we need reliable methods to predict significance of these variants in disease. So the question we are asking, can we use family data in low or no data situation for a protein of clinical interest? In our work, we have come up with an approach to train a family specific predictor where we have collected variants for a particular family. In this work, we have used NAGLU protein as a case study. Now let's look at what NAGLU is. NAGLU, it stands for N-acetyl alpha-glucosaminidase. NAGLU is a protein in human that is encoded by NAGLU gene. NAGLU degrades heparan sulfate by hydrolysis of terminal N-acetyl D-glucosamine residues in N-acetyl alpha-D-glucosaminates. Any defects in the NAGLU gene can cause accumulation of heparan sulfate, leading to disease like Sanfilippo B disease. Sanfilippo B disease is a neurodegenerative disorder which results in intellectual disability, eventually leading to dementia and behavioral issues. There are many missense mutations associated in NAGLU that are uncharacterized. Our work demonstrates how these mutations can be characterized in the absence of enough experimental data for NAGLU protein. So here we have collected family-specific variants of the NAGLU protein, which are compiled from different databases such as Humsavar, ClinVar, Nomad, Pompe disease mutation database, and LOVD. We trained our predictor on three different datasets. The first dataset we call carbohydrate processing enzymes dataset. This dataset consists of variants from 16 proteins, including NAGLU protein, belonging to carbohydrate processing hydrolysis of lysosomes. A total of 1802 variants are present in this dataset, which includes 95 variants from NAGLU protein. The proteins in this dataset processes similar substrates and co-located in lysosomes. The second dataset is hydrolysis dataset. This dataset has variants from 32 proteins of the hydrolase superfamily, which includes proteins from carbohydrate processing enzymes as well. A total of 1988 variants are present in this dataset, which includes 95 variants from NAGLU protein. The third and the final dataset is a control dataset. This dataset contains all the protein variants from hydrolysis data except NAGLU. A total of 1893 variants are present in this dataset, which does not include 95 variants from the NAGLU protein. Here, I will go through the model architecture and the features that we used for training. We trained a transformer-based neural network model on variants of genes functionally related to NUGLU gene. These variants could come from gene family or superfamily. Our method combines log odds score from zero-shot predictions as a feature along with embeddings from ESM2, a protein language model, as features to train a supervised model on variant data. For representing proteins, we considered a residue level representation. For residue level representation, we extracted amino acid level features using ESM2 language model only at the site of mutation for both wild type and the mutant protein, and then calculated the difference. Furthermore, we trained a model where we also used PCA to reduce the embedding size from ESM2 and use these reduced features as input along with zero-shot prediction as feature to train the supervised model. This slide shows the complete model architecture using residue level representation. Here in this slide, the table shows the prediction performance for NUGLU VUS. In order to test our model against existing state-of-the-art models, we created a test set which consists of 38 pathogenic and 126 benign variants from NUGLU gene. We made sure this test set does not overlap with variants in our training set. As shown in the table, the average precision score for all our models is substantially better than the state-of-the-art predictors SNAP2, Polyfin2, and SHIFT, and is slightly better than MutePred2. Also, we can see from the table that the models augmented with family data have a better balanced accuracy and average precision score compared to the zero-shot model. This goes to show that family variant data can serve as a proxy training data if gene of interest has no or low data problem. So in conclusion, the supervised model leveraging ESM2 embeddings as features along with zero-shot log odds feature captures traits specific to the protein family and helps in improving the predictions. Thus our model captures both generic and contextual property of the proteins resulting in increased performance. Our work here highlights that models trained on family or superfamily data can be used to predict variants for proteins that have low data or no data problem with a good confidence. Lastly, this method is applicable to other protein families as it does not need any feature engineering and only requires collection of family-specific variants for training. I would like to thank my colleagues Dinesh Joshi and Rakshanda Sajid for their inputs. And I would like to thank my mentors, Dr. Sadhna Rana and Dr. Rajgopal Srinivasan, for this opportunity and their guidance. And this brings us to the end. I would like to thank everyone for the time and attention.